Hi friends, this is Anand Science Academy. In this video, we are going to discuss the topic Chromosomal Theory of Inheritance from the chapter Principles of Inheritance and Variation. Let's see what is this chromosomal theory of inheritance. Mendel published his work on inheritance of characters in the year 1865, but for several reasons, it remained unrecognized till 1900. Firstly, communication was not easy in those days and his work could not be widely publicized. Secondly, his concept of genes or factors, in Mendel's words, as stable and discrete units that controlled the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900, three scientists, Hugo de Vries, Carl Correns, and Erich van Schermach, independently rediscovered Mendel's results on the inheritance of characters. Also, by this time, due to advancements in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes, that means colored bodies, as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's laws. Recall that you have studied the behavior of chromosomes during mitosis and during meiosis. The important things to remember are that chromosomes as well as genes occur in pairs. Two alleles of a gene pair are located on homologous sites on homologous chromosomes. This you have studied in 11th standard. You can just recall that. With this figure, let us just recollect meiosis. There are 4 chromosomes. During meiosis 1, the actual reduction happens. The 2 daughter cells are formed after meiosis 1. In those 2 daughter cells, we have only 2 chromosomes, and these 2 cells undergo meiosis 2. As a result, 4 cells are formed. Each has 2 chromosomes. At the end of meiosis, 4 daughter cells are formed with half the number of chromosomes. There is a table in our NCERT reader showing a comparison between behavior of chromosomes and genes. There are 3 points. Below this table, there is a question. Which column represents chromosome? Which column represents genes? And the reason is also asked. Here, the word independent can be used for genes only, as the chromosomes are always independent. Genes that show linkage are not independent. What is this linkage? Let us discuss that in the next video. One gene need not be separated from another independently when they are linked on the same chromosome. That means when the 2 genes are located on the same chromosome, we need not expect that they have to get separated independently. Thus, Column A represents chromosome and the column B represent genes. During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently of each other. You can see that in the figure. To understand this, compare the chromosomes of 4 different colors in the left and right columns. There are 4 different colors: orange, yellow, green and red. In the left column, orange and green is segregating together, but in the right hand column, the orange chromosome is segregating with the red chromosomes. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies Drosophila melanogaster, which were found very suitable for such studies. This name of the insect, Drosophila melanogaster, is a frequently asked 1-mark question. Why Morgan has selected this Drosophila particularly? These are the reasons. This is also a frequently asked 3-mark question. These fruit flies could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks. Single mating could produce a large number of progeny flies. There was a clear differentiation of the sexes. The male and female flies are easily distinguishable. It has many types of hereditary variations that can be seen with low power microscope. Because of these reasons, Morgan has selected this Drosophila melanogaster or fruit flies for his experiments. What are the characters Morgan studied with these fruit flies and what are the results he obtained? All these topics let's discuss in the next video.